Good evening, everyone. Welcome in today's class. So, as we say in the starting, let's crack NEET PG together. So, let's begin with the class. The topic that we are going to discuss today is rDNA technology. Just a second. Before beginning with rDNA technology, let us have a small introduction about Unacademy. We're talking a little bit about Unacademy. Very short introduction. Unacademy is one of the India's largest platform for the preparation of NEET PG examination. And on Unacademy, you will find one of the India's best educators. So these are some of the picks of the best educators of the Unacademy. So you can be easily mentored by these eminent faculty members at Unacademy. I hope my voice is audible to everyone. Those who are live, they can just answer me in the chat box if they can hear me clearly. Then, whenever there is a kind of preparation for the any kind of competitive examination, as I always say, that what you need to do is you need to continuously evaluate yourself that whether you have learned the concept or not. So test series can be very much helpful in these kind of things. And with Unacademy, this is possible because there are free mock tests and weekly tests with Unacademy. You can just go and attend the test, and you can retake the test as many number of times you want to. You can evaluate yourself. Then it is very flexible way of learning because you can just go to the Play Store and download the app, and once the app is downloaded you just need to make your account and you are good to go. Then there will be no botheration about being physically present in the offline class and time management if you are not in the town, nothing. It is very flexible. Wherever you are, you can just continue your preparation. In language of your choice, there are many educators who even teach in the regional languages, so they are helping the students out in preparation too hard, so you can very well make the most out of the available content. Then unlimited practice is another added benefit, that the videos, if once they are recorded, they can be rewatched, and if the quiz once conducted, it can be retaken. So these are the very important benefits with online learning and with Unacademy. Then there are some students who feel whether we will be feeling like classroom or not. So it is exactly classroom like feeling, because you can chat with your educator, you can ask your doubts, that what type of problems you are suffering while preparation. So you can chat with your educator very well, and they'll be very much willing to answer. Then there will be interactive polls. You can just answer the questions which your faculty member they are asking, and you will also come to know that which are the overlapping options, how many students they are confused in the same concept, and faculty members they'll be explaining out each and every option in detail, so you can also understand why not the option which you have chosen. Then learn anytime anywhere, because it is an online kind of learning. You need to be so you can learn anytime anywhere. If once you have missed an online lecture, you can very well take the recorded session. So very much flexible it is. Then with Unacademy, there is another a great feature with Unacademy is they have got this plus subscription. And on this plus subscription, you will be getting all the 19 subjects and all the topics of these 19 subjects recorded and live lectures. For this plus subscription, you need to pay a very pocket friendly amount, and by that you will be eligible to watch all these. And time to time they conduct the short term courses and crash courses, so it will be very much beneficial for you for preparation of upcoming exam and to have the knowledge of what kind of exams are being conducted. So these things are possible with plus platform, and for this plus platform the amount is very much pocket friendly, and if you're going for a year or a 2 yearly program, then it is very reasonable. It is very reasonable, pocket friendly, and you can make the most out of the topics available and the videos available in this duration, like 2 years or 1 year. subscription. Now in this subscription also you can get an additional 10% discount if you are using my referral code, that is FB01. So just use my referral code, that is FB01, and avail an additional 10% discount. So don't forget my referral code and get benefited out of it. Right, so let's move further in our class, that is recombinant DNA technology. So this recombinant DNA technology is a kind of molecular technique. So the topic name is molecular technique, and that is your recombinant DNA technology or rDNA technology. So there are many terms used for rDNA technology, and they all remain means the same. If someone is calling genetic engineering, then it is rDNA technology, or someone is saying gene cloning, then also they mean rDNA technology. So cloning and rDNA technology, these all things are same, right? So we'll be talking about this topic in detail in today's class. Everything we'll be talking, so we'll be dealing it under the following heads, that is definition, steps, tools which are used or molecular tools, and lastly application. So these all things we are going to in today's class. So it is one of the most interesting topic and quite important these days, because this is a new thing and it's a biotechnological concept which is embedded in biochemistry. Right, so we'll be talking about rDNA technology in today's class, that how this recombinant DNA is prepared, what all things are needed. Right, so let's move further. We'll be starting with the first point, that is the definition. Definition. So what is the definition of rDNA technology? Manipulation of genetic material to achieve a desired goal in a predetermined way. So what is it? It is manipulation of genetic material, because genetic material will be changed. So manipulation of genetic material, directly we'll be altering the genetic material only, rather than giving the drugs or anything, directly genetic material, so that it will be embedded in the genes to achieve a desired goal, and that will be in predetermined manner. So this is our rDNA technology or genetic engineering. Now, if this is the definition, that we need to manipulate the genetic material, we are saying that we need to manipulate the genetic material. So this manipulation must be done in various steps, because if we are talking about genetic material, we need to deal with the genetic material. So it needs to be isolated, and everything related to genetic material will be done. So what all things will be done in manipulation of genetic material, those all things will sum up in the six main steps. Six main steps. So we'll be dealing it like that. So I'll be just first of all telling you what all those steps are, and then we'll be going and seeing each and every step in detail. So the first step is isolation of desired gene. The gene of interest will be isolated from an healthy individual. Or if we are just looking for a case study, or we want to do a mutation study, so we need to first of all isolate the gene, right? So isolation of desired gene, then isolation of vectors, isolation of vector, then vector means it's a carrier. Vector, the general term for is carrier. The carrier of the required genetic material or desired genetic material called as vector. Then production of chimeric DNA. Production of chimeric DNA. What is chimera? Whenever we talk about hybrid, we call it chimera. So production of chimeric DNA. Then introduction in hosted cell. Introduction in host cell. Like if we are saying that we want a gene or we want a product of a particular type of thing, so we need to take the production out of it, or we need its expression. So introduction in host cell. And multiplication and selection of cell with rDNA molecule. Like there will be so many cells which will be undergoing changes. So those cells which have engulfed the proper recombinant DNA or a proper chimeric DNA, those cells need to be selected. So that is multiplication and then selection of cells with rDNA molecule. Right, those cells which contain the rDNA molecule. So I'll be telling you what are the techniques for each and every step. So this is just the outline. So multiplication and selection of cells with rDNA, right? Then next thing is expression of gene to get the desired product. Expression of gene to get desired product. Now if we are aiming to get a particular type of production, so we need to get the expression of gene. So that is the sixth step, that is expression of the gene which we are incorporating, right? So these are the major steps under which we will be doing our whole rDNA technology. We'll be doing each and every step in detail. So let's move further towards the next step. So what is the heading of this step? That is isolation of desired gene. This desired gene is also called as gene of interest. Gene of interest, whichever gene we are interested in to see what is its production or to see whether it is mutated or not. So that will be our desired gene, whether a gene has been mutated, now its product is a active product or it is not an active product, it's an inactivated protein. So we need to isolate the gene. So from where are we going to isolate the gene? We'll be isolating the gene definitely from cell, right? Cell is the main source where we'll be getting the gene. So cell, then we'll be hydrolyzing the cell because in cell the genetic material will be present in nucleus. So we need to hydrolyze the cell wall of the cell. So it will be undergoing some enzymatic treatment. There will be use of some detergents for breaking the cell membrane and cell wall. And then we will be also using certain kind of phenols and proteases and ribonucleases, right? So we don't want any kind of protein, so we'll be using proteases. We don't want any kind of RNA, so we'll be using ribonucleases. But we want DNA, the genetic material. This is what is in demand, a particular gene. So what we will do, we will just eliminate out all of the proteins and all of the RNAs with the help of proteases and ribonucleases. And when this will be done, we will just perform the ultracentrifugation of whole thing so that the genetic material can be obtained easily. So after ultracentrifugation, we will be adding chilled chilled ethanol. And when we will add the chilled ethanol in the centrifugation tube, what will happen? The DNA will precipitate out. And when the DNA will be precipitated out, it will be very much easy to obtain the DNA from that mother liquor. So DNA has been precipitated out, good, fine. Now, do we need whole cell, whole genetic material, whole genome? No, we don't need whole genome. It is very bulky. We just need a gene. If this is the whole genetic material, and let us take another color for making gene of interest, and this is our gene of interest. So we cannot just isolate this gene of interest directly from the cell. We need to isolate the whole genetic material and then we can cut out the gene of interest. So we just want this red color thing, this gene of interest, right? So what we will do, we will perform the restriction, we will apply the restriction endonucleases so that they can cut at the places where we want. So the genetic material, it has got certain sequences where a specific type of endonuclease will work. Endonuclease is an enzyme which cuts the DNA at the interior. So they are sequence in specific enzymes. So we'll just identify that type of endonuclease which can cut a particular place in the whole genetic material, right? So restriction endonuclease will cut the genetic material and we will be obtaining small fragments of DNA. And these small fragments of DNA will contain the gene of interest also. So from all these, how will they be differentiated? Then another technique will be applied, that is electrophoresis. Electrophoresis. In electrophoretic unit, these sets of small fragments of DNA, they will run at a different pace and they will be occupying different space in the slab. So there will be a marker slab also, and wherever the gene of interest will collide or it will show the same level, it means that is our gene of interest, right? And again, to make it 100% that yes, it is our gene of interest, what next will be done, that will be southern blotting. Southern blotting is simply nothing when this whole sequence which is set on the electrophoretic unit, there will be a nitrocellulose paper which will be kept over it, and all the bands will now come on the nitrocellulose paper. Now when the bands, they are arranged on nitrocellulose paper, they can be further applied with the probe, and if probe it is binding with a certain sequence, it means that is the gene of interest, or that is our desired gene. So this is how isolation of desired gene occurs, that is with the help of a cell, or the starting material is our cell. So this is the very first step, that is isolation of desired gene or DNA, right? And we have just cut the genetic material with the help of endonuclease. So the genetic material which will be cut with the help of endonuclease, it will have sticky ends. That is some of the fragment which is at the ends of the genetic material, they will have free ends. That is, they won't be in double stranded form, but they will be in single stranded form. So these single strands hangings will also called as sticky ends. Sticky ends. Sticky end DNA molecule. So this type of sticky end we get with the help of restriction endonuclease enzyme. So when we are saying that they are sticky ends, it means they can bind with some other complementary sequences. If a complementary sequence is having the same kind of sticky overhang and it is having the complementary sequence, so they can easily make the bond with them and they can just double, or they can just make a complete double strand over the place where they are hanging also. So this is the concept of sticky ends, right? So with the help of endonuclease, we have got these sticky ends, right? Now this was our first step. Now moving to the second step, that is your isolation of vectors. Isolation of vectors. Now, what are these vectors? They are simply the carriers. They will simply carry the gene of interest into the host cell. So, vectors mostly they are derived from bacteria, or they are derived from yeast. So, whenever we are isolating some kind of vector from bacteria, what we will do? We will take the bacterial cell. Now, this bacterial cell will have some kind of cell wall around it, so that needs to be broken down. So it will be broken down with the help of enzyme, EDTA and lysozymes. This combination will be added in a suspension of bacterial cells, and for removing any other kind of thing, sodium lauryl sulfate, sodium lauryl sarcosinate will also be added in the solution. So when all these things will be added, the plasmid will be freed, and plasmid can be isolated from the whole thing with the help of ultracentrifugation, and finally with the help of electrophoresis, which we have seen, just like isolation of desired gene. Now when we are saying that this bacterial cell will give us vector, and the vector over here is plasmid. So let us talk a little bit about plasmid, that what is this plasmid, right? So this plasmid is nothing but extra chromosomal material of bacterial cell. It is not required for the survival of bacteria. If this is the bacterial cell and this is the genetic material of bacteria, this is our plasmid. This is plasmid. Now it is not required for survival of bacterial cell, but it will be present and it will be giving bacteria some special quality. Special quality. If there is a plasmid, it will make the bacterial cell well equipped with certain type of characters, right? So now we have obtained the plasmid, and I have told you a little about plasmid, and in further slides we will be also talking about the tools of genetic engineering, and in that we'll be talking about vectors, and I'll be telling you the detail of vectors and specifically plasmid. So here plasmid we have obtained. So our gene of interest was having sticky ends, right? And our plasmid over here, it will also have sticky end when it will be cut with the help of restriction endonuclease. So this is our plasmid and it is having sticky ends because some of the overhangs, and they both are cut with the help of same endonuclease. When it will be cut with the help of same restriction endonuclease, both will have the same sticky end. So now they can very easily join with each other. So here is our plasmid with the sticky end, and here is our gene of interest with sticky end. So the very next step of the process of recombinant DNA technology is formation of chimera. Formation of chimera or chimeric DNA, and it is also called as DNA hybrid. Why it is called as DNA hybrid? Because the vector is obtained from bacteria or yeast, and the gene of interest will be obtained from the desired organism, from whomsoever we want the product to be formed, right? So the formation of chimeric DNA. So very simple, this is our gene of interest with overhangs. This is our gene of interest, and this is our plasmid with the overhang. So when they both will combine, they will form a chimeric DNA, right? This kind of, we'll just take another color for the same. So it will be circularized and will be now complete. So this is our chimeric DNA, and this process occurs in the enzyme DNA ligase. DNA ligase is responsible for this activity, and it will be ATP dependent activity, right? So formation of chimeric DNA, that is, it will contain a part of plasmid and a part of the host gene. So that is the gene of interest. Now this is how our chimeric DNA is formed. Now coming to the next step of the recombinant DNA technology, that is introduction in host cell. Introduction, introduction in host cell. Now the recombinant material that we have made, that is our chimeric DNA, it needs to go inside the cell and it needs to do the process. So there will be various gene transfer methods through which this is the material of bacteria. Usually the host cell that is taken is bacterial cell. Bacterial cell is usually used as the host cell. So why is a bacteria more preferred over another thing? Because bacterial cell are very easy to multiply, and they are very efficient in production of whatever thing we want to be produced. So this is bacterial chromosome and this is our chimeric DNA, right? This is our chimeric DNA. We are just writing cDNA, but it is actually chimeric DNA. See, so bacterial cells, they'll be preferred as in host cells. So while the host cell, how will the host cell be uh, prepared, or how the host cell will take the genetic material? What will happen? There will be various types of gene transfer methods. Gene transfer methods. So with the help of various gene transfer methods, the genetic material which we have just prepared or manipulated, it will be introduced into the bacterial cell. And we'll be talking about some of the names of gene transfer method in the coming slides. I'll be telling you about 3-4 types of gene transfer methods as well. Now, the very next step is multiplication. Multiplication in host cell and selection of host cell, and selection of host cells with chimeric DNA. With chimeric DNA. Right? So now if this is the bacterial cell and it has got its own genetic material and it has got this chimeric, has got the chimeric DNA as well. And there will be some of the bacterial cells which will remain untransformed and they will just contain its own genetic material. So we need to select the cells which have got the chimeric DNA as well. So how are we going to select these cells? There is a very special feature with the vector, that these vectors are contain some of the genes for antibiotic resistance. The plasmids, they themselves in nature contain R plasmids. R plasmids. So these R plasmids confer some type of antibiotic resistance. Various types of antibiotic resistance genes are present in various types of R plasmids. So they can be used as a vector. So that's why also plasmid is preferred, because it has got antibiotic resistance gene. So it is very easy for them to be selected. So are plasmids. Then there will be use of probes. Certain probes, they can also prove whether a particular sequence is present or not. They will be applied on the cell, and if the cell it starts glowing, it means yes, it has got the desired gene. So the multiplication and further selection. So multiplication is easy because it's a bacterial cell, it will be multiplied, and your vector will also multiply in the bacterial cell well, because they'll be belonging to the same species. And then finally there will be formation of chimeric DNA and its selection. Its selection is done with the help of antibiotic resistance genes and probes, right? So this was the fifth step. Now talking about the very last step of this process, our DNA technology, that is expression of gene to get the desired product. Let me take a pen. So expression of gene. Expression of gene to get desired product. Now the product can be protein, it can be a kind of growth hormone, anything. So this is the bacterial cell, this is its genetic material, and this is the vector. So definitely if the gene is there, it means it will be expressed, and it will be expressing in the term of product. This is the very sixth step of the process, expression of gene to get the desired product. So if it is forming the product well in the environment, it means that fine, the process is successful for this rDNA technology, and this host cell mechanism can be further used in future for the production. And these type of cells, they'll be cloned many number of times and they'll be preserved for further usage, right? So this is how the rDNA technology it occurs. This is the basic step. This is the gist of the whole process. Now we need to know what are the molecular tools, what are the enzymes which are used, and there are many questions which are asked on this also. So let us talk about the molecular tools. Very important. Molecular tools. So molecular tools, it will be made up of various types of enzymes, and we'll be just seeing and dealing them one by one, right? So in the process, the first enzyme which we are talking since starting, the first enzyme, that we are talking is restriction endonuclease. So these nucleases, they are of two types, that is exonucleases and endonucleases, right? So endonucleases are important over here because they'll be cutting the genetic material somewhere in the middle, and exonuclease, they'll be cutting the ends of the genetic material, right? So endonuclease are quite important when we talk about genetic engineering because they are also called as molecular scissor because they can cut at a very specific site. They are sequence specific in nature. So they are also called as molecular scissor, right? So molecular scissor. Also, we'll write the name, that is restriction endonuclease, right? Now, restriction endonuclease is responsible for cutting the things. There are three types of restriction endonucleases. Restriction endonucleases 1, 2, and 3, right? So, the restriction endonuclease 1 and 3, they are ATP dependent. ATP dependent, and they make a cut away from the recognition sites. Cut away from recognition site. Now, when it makes a cut away from recognition site, its importance it is quite decreased. It won't be of much importance because we want the thing to be very, very precise and we want it to be further used. So if it is making a cut away from the place, it will be of no much use. So which kind of endonuclease is important in that case? It is restriction endonuclease 2 will be important. And what are the features of this restriction endonuclease? Let's highlight the feature of restriction endonuclease 2. What does it do? It doesn't require ATP. It is not ATP dependent. It doesn't require ATP for cutting. The second thing that is so good about it at the cut at the recognition site. Recognition site, right? It will make a cut at the recognition site. Now, when we are saying that it will make a cut at the recognition site, so what is this recognition site? Recognition site. Which place will be called as recognition site? The recognition site. And what will be the length of recognition site? So recognition site can be 4 to 6 nucleotide long, right? And usually recognition sites are palindromes or palindromic sequences. Very important. What is a palindromic sequence? That is, if it is read from a left direction or if it is read from right direction, will read same. But when we are talking about genetic material as in palindrome, how will it be called as palindrome? Just imagine the sequence is GAA, TT, and C. So the complementary will be C, TT, AA, and G. So now just read it from this 5 dash to 3 dash, and here 5 dash to 3 dash. GAATTC and GAATTC. So this is a kind of palindromic sequences. So palindromic sequence can also be understood with another example of a word, that is madam. MADAM. If you are reading it from left or you are reading it from right, they both will appear same. So this is the importance of molecular scissor, that is your palindromic sites, and they are mostly 4 to 6 nucleotide long, right? So they are not very long, but they are just 4 to 6 nucleotide long, and they are palindromic in nature. That is, they will be read same in both of the direction. Now, coming to the next thing about restriction endonuclease, that is how is it named. So the restriction name and restriction endonucleases, they are usually named like this. So here E stands for genus of the organism from which it is isolated. O stands for the species from which it is isolated. R stands for the name of the scientist who actually gave the vector, that is Roderick's name of scientist, and one tells you about the order of discovery. Order of discovery, right? So this is how a restriction enzyme is named. So if in question you are asked about anything, that what is the first alphabet in the name of the restriction enzyme, what does it mean, and the numeral, what does it mean? So numeral is usually about the scientist, about the sequence of its discovery. This numeral is usually about the order of discovery. When was it actually discovered, right? Then let us talk about eco Ri or eco R1. So what is the use of this eco R1? If it is present naturally in nature, in bacteria or in E. coli, what is its use in E. coli? Like if we are using it for manipulation, there must be some of its use. So it's a kind of enzyme, right? It's an enzyme. So enzyme, and that is also an endonuclease. It means it is cutting somewhere in the middle. So what does this do? It restricts the growth of viruses in bacteria. It restricts growth of virus in bacteria. This is its major function, right? And if we are saying that it is cutting the palindromic sequence and it is also producing the sticky end. So just look at this sequence. If it is cutting somewhere here and it is cutting here, so it will be producing sticky end. That is, this portion will go together and this portion will go, this portion will go together. So like this, it will be producing sticky ends. And there can be a question, or there are PYQs which I have seen, that what is the use of restriction enzyme in bacteria itself. So the use of restriction enzyme in bacteria is restriction the growth of the invading viruses in bacteria. There will be infection of virus in the bacteria, that is called as phage. The virus which infect bacteria, they are called as phages. So there will be restricted growth of phages if that particular bacteria has got restriction enzyme of a particular type, right? So we have talked everything about restriction enzyme. The second enzyme of the story is ligase or DNA ligase. So we have just seen from the starting when we were talking about the steps, the DNA ligase, it is responsible for making a chimeric DNA, right? And many other places also it will be needed. But the thing that we have seen in previous slides is it is used in forming the chimeric DNA. And this DNA ligase is also called as molecular glue. The restriction enzyme was called as molecular scissor. This DNA ligase, it is called as molecular glue. And this molecular glue or DNA ligase, it is ATP dependent. ATP dependent. And the restriction enzyme which we were using widely, that is our restriction endonuclease 2, this is non-ATP dependent. There are many PYQs which are asked on this, that restriction endonuclease 2 is ATP dependent, ATP not dependent, or it may require ATP, none of the above. So it is very important to remember which of the enzyme is ATP dependent and which enzyme is not ATP dependent. So remember, DNA ligase is your ATP dependent and it is your molecular glue. So that can be very easily remembered because the name is very simple, that is molecular glue, right? And if it is a molecular glue, it means we also need to know what kind of bond it is making, right? So it is making phosphodiester bond. It is making phosphodiester bond, right? So this is about our DNA ligase. Now let's move towards the other enzyme, that is polymerase. So polymerase are always needed for the regular replication of the genetic material, whether it is the main genetic material or if it is the extra chromosomal material. Now fourth, we need alkaline phosphatase. Alkaline phosphatase, the enzyme that is needed. Now after that, we also need reverse transcriptase. Reverse transcriptase. reverse transcriptase. So why is this reverse transcriptase needed? Because many a times it won't be possible for us to isolate a gene directly. So we can just isolate the transcript which has been formed from the gene. So whenever if we are isolating mRNA and we just want the gene for the production of this kind of mRNA, we will perform the restriction transcription of this mRNA and we will be getting the cDNA, that is the complementary DNA. And this complementary DNA will be introduced in the genetic engineering pathway and it will be further used for the formation of rDNA. Right, so there is a very important use of reverse transcriptase. There are various libraries which are called as cDNA libraries. Like there are genetic libraries or whole genome libraries, and there are another libraries which is called as cDNA library. Right, what is the importance of cDNA library? The whole genome is not utilized for formation of protein, right? Only some of the part of the whole genetic material is used for the production of protein. So all the expression libraries where actually the thing which is making a whole phenotype is present is called as cDNA library. And if whole genotype we want to preserve, then we'll be going for genomic libraries. And if we just want the expression libraries, okay, in the lifespan of a particular organism, what all genes are into work play, we'll be going for cDNA library. Right, now the last enzyme of this category which we are discussing, that is the enzymes. The last enzyme of the category is RNase H. RNase H. What is the function of RNase H? If those students who are attending now and they have seen my molecular uh, genetics or molecular biochemistry videos, they can easily remember that RNase H can disrupt the DNA RNA interaction. The DNA and RNA, whenever it is present in a close association or wherever there is a one strand of DNA and there is another strand of RNA, RNase H disrupts this type of bonding. Right? So this is about RNase H. RNase H, it disrupts the DNA RNA hybrid. Right, so just remember the use of RNase H. Now are done with the first molecular tool, that is the enzyme, and let us move further towards the next molecular tool, that is vector. Quite important because it is highly asked in exams. Vectors, what is a vector? So we are always reading that vectors are the are those type of material which are actually responsible for transport. So they can be also called as transport vehicles. They are the transport vehicles. Now, transport of what? Transport of desired gene. Transport of desired gene into the host organism. And when we were doing the initial steps, I told you that I'll be talking in detail about vectors. So let us start the detailing of vectors. The first vector that is used is plasmid. Plasmid as in vector. So this plasmid, what is it, this plasmid? This is an extra chromosomal material which is not responsible for the survival of bacteria. And if it is present, it gives it some specific or special qualities. So what is the structure of plasmid? It is stranded, double stranded extra chromosomal material. Extra chromosomal material, right? And what is the other important thing about this plasmid is that it is responsible for independent replication. Independent replication. Right? These are the two qualities. Now, whenever we'll be talking about vectors, we will be always talking about that how much genetic material a particular kind of plasmid can carry in itself. Like if it's a vehicle, it cannot carry unlimited amount of anything. Like a auto rickshaw can take 3 to 4 people and a bus can take not many number of people and car and a scooter, like that. If we are calling it vehicle, then we also need to determine how much amount of genetic material is it carrying. So like plasmid, it will be carrying less than 10 kb of the genetic material, right? Then we talk about bacteriophages as in vector. bacteriophage. Bacteriophage as as the vector. Then let me just write down the names, and then I'll be telling you what mount it will be carrying. Bacteriophage, then comes is cosmet, then comes yeast artificial chromosome, then is your bacterial artificial chromosome, and lastly mammalian artificial chromosome. These all are kind of vectors, and the amount that they can carry will be just seen. Bacteriophage, it can carry around 10 to 20 kb of genetic material. This genetic material means what? Our desired gene, right? This and rest other thing, it is its own bulk. If its own bulk is something, so that is altogether a different thing. The bulk of gene of interest which it is carrying, that we are writing over here, that we are highlighting. So cosmet, it can be 35 to 50 kb. In yeast artificial chromosome, it can carry a very large amount of desired gene, that is 1 MB. Bacterial artificial chromosome, it can carry less than 300 kb, and mammalian artificial chromosome, it will be carrying around 1 MB of genetic material. This MB means mega base pair, okay? And kb means kilo base pair, right? So mega base pair and kilo base pair. Kb, kilo base, kilo bases, and MB is mega bases, mega bases, right? KB and MB, kilo bases and mega bases, right? So mammalian artificial chromosome and yeast artificial chromosome, they will be carrying different amount of genetic engineering, and they will be carrying highest amount of genetic information. So we have seen vectors, how many types of vectors? Plasmid we have seen, then we have seen bacteriophage, then we have seen about cosmid, yeast artificial chromosome, bacterial artificial chromosome, and mammalian artificial chromosome. Right? Let's move ahead. Now we have done complete thing about the vectors. Yeah, one point I need to mention in vectors is about this plasmids. If plasmids, we have talked about one of the plasmid, that is EcoR1. There are many other types of plasmid also, like PUC, plasmid isolated in the University of California, that is PUC 101, and 101 just tells its sequence of discovery. Then there is another plasmid, its name is PBR 322. P stands for plasmid, B and R stands for the scientist Bolivar Rodriguez, and 322 is the sequence in which it was isolated or identified, right? So these are some of the example of plasmid. Fine. Now moving to the next molecular tool of the story is the host cell. How will one identify that, okay, this can be the host cell? In this host cell, we'll be incorporating the gene of interest for the further thing. So host cell, the host cell, preferably they are prokaryotic in nature. They will be prokaryotic. Why prokaryotics are preferred? Because in prokaryotes there will be rapid multiplication. They are preferred for rapid multiplication, right? Then if we are saying prokaryotic and they are very much helpful in rapid multiplication, so what are the examples of prokaryotic? That is E. coli and Bacillus subtilis, right? And if prokaryotes is the most preferable one, sometimes there will be eukaryotic host also. So all things can be used as an eukaryotic host. In eukaryotic host, fungus can serve as eukaryotic host. Then plant cells can work as the host cell, or mammalian cell can also work as the host cell. And in some of the cases, Saccharomyces cerevisiae will also be used as the host cell. So if we are saying that bacteria, they can multiply fast, and we are seeing so many importance of prokaryotic, that prokaryotic are much better because they can grow in the minimal requirement, then why eukaryotic? Is there anything important related to eukaryotes? Because when we are saying that prokaryotic are better, so there are some of the cases where the protein which is formed, it needs to undergo post translational modification. It needs to undergo post translational modification. So whenever there will be a need of post translational modification, that is possible only in case of eukaryotic cells. So in those cases, eukaryotic cells will be used as in host cells, right? So in case of post translational modification, so never forget this thing, that why eukaryotes are preferred in some of the cases. So there can be a PYQ, that why some of the eukaryotic cells are used as the host cell. Post-translational modification, post-transcriptional modification, replication system, none of the above, all of the above. But now when I've told you that it is the post-translational modification, you are going to take post-translational modification. That is when the protein is formed, it needs to undergo various types of modification for becoming a completely active protein molecule. Right? Understood the thing well? Now, the last molecular tool that we are going to see is gene transfer techniques. Gene transfer techniques. So, gene transfer techniques, as I was telling you, there are various types of gene transfer techniques. We'll be just writing the name of each and every gene transfer technique, and I'll be outlining the processes. Transformation. Transformation. Naturally, if the bacterial cell, they are lying in the environment, they can take up the genetic material which is lying in the environment. So in a media, we can just put the gene of interest and we can leave the bacterial cell so that it can spontaneously absorb it. So that is transformation. Then there is another process, that this is also a natural based process, that is transduction. What happens in transduction is the bacterial cells, they are usually infected by some kind of viruses. So the virus, if it virus particle contains the gene of interest, then it can also deliver the gene of interest inside the host cell. So this gene transfer, yes, gene transfer where we are doing this gene transfer from your vector, how the vector will go inside the host. This is the question, how the vector will go inside the host. So the first way it can go is transformation. The gene of interest will lie in the environment and it will go inside the bacterial cell. The second way it can do is through transduction. The viruses which can naturally infect the bacterial cell, those viruses will be inserting their genetic material in the bacterial cell. And then third process through which it can go inside the bacterial cell is through electroporation. Electroporation. Can you see the term? It is so interesting. Electro. Electro means electricity. With the help of electricity, there will be formation of pores in the bacterial cell, and with the help of these pores, there will be just incorporating the genetic material in them. If these are the bacterial cells and in the outer environment, these are the gene of interest, they are lying. So here will be a positive electrode and here will be a negative electrode, right? So when a positive electrode and a negative electrode, there will be a flow of current. There will be a transient formation of pores in the bacterial cells. Transient pores in the bacterial cells, and with the help of these transient pores, the genetic material can go inside it. And the pores, they are made by a very feeble amount of current. When the current is very feeble and due to the flexibility of the cell membrane, the cells can take up the genetic material very easily. So this is our electroporation, electricity and formation of pores, right? And then next thing is gene gun. Gene gun. So the word itself is telling that there will be some kind of unit and there will be a platform, and on this platform the cell will be lying and there will be just a shoot of the gene on this cell, and the shoot will be of a particular strength so that the genetic material can go inside the host cell. So gene gun, there's a detail about this gene gun that we can discuss in some other class, that is the gene transfer techniques. right? So gene gun. Now the fifth technique is liposuction. Liposuction. What is liposuction? Lipo means lipid and faction means infection. Right there, the cell membrane is lipid in nature. Let's draw here. The cell membrane is lipid in nature. If the gene of interest it is entrapped in a molecule which itself is lipid in nature, then the cell membrane and the lipid molecule they will show likability towards each other, and because of this likability this lipid molecule will fuse with the cell membrane and the genetic material will be incorporated into the cell. This is the process of liposuction. Lipo means lipid molecule. It will be kind of a mesyl molecule like this, hydrophobic interior and hydrophilic exterior, and the genetic material will be trapped inside it in the mesocyl, and it will be just combining with the cell membrane of the host cell, and it will be delivering its genetic material in the host cell. So we have seen in gene transfer techniques, we have seen transformation, then we have seen transduction, then we have seen gene gun, and we have seen liposuction, right? So these are some of the gene transfer techniques. Gene transfer techniques. So gene transfer techniques are very much important for transferring of vector into the host or transferring the gene of interest into the host cell, right? So we have discussed almost everything regarding the technology. So after knowing the whole technology, what we need to know, if we know that okay this is a certain process, what will we need to know after that? We need to know what are the applications. What are the applications? So these applications are in context to the medical uses. So we'll be seeing the application of rDNA technology. Very important, because this is the actual thing which we need to know, right? Application of the thing. So we'll be dealing with the application, right? So let's start with the application. The first application, let me just change the color, right? So those all who are attending the class live, they can just write in the chat box if they are understanding, and if they are getting the concept, they can just write in the chat box. So first application is production of therapeutic drugs. Production of therapeutic drugs. So production of therapeutic drugs, let us deal with a very important example, that is insulin. Insulin hormone production. Insulin hormone production, right? Insulin hormone production, it is used in the it is used in the it is used in the treatment it is used in the treatment of diabetes mellitus, right? Insulin hormone, it is used in the treatment of diabetes mellitus. Just a second. Diabetes mellitus. Diabetes mellitus. It is used in the treatment of diabetes mellitus. So the insulin hormone, it is made up of two chains. It is made up of two chains, that is A chain and B chain. It is made up of A chain and B chain, right? So the gene for A chain will be inserted in an E. coli, and the B chain will be inserted in another E. coli. So E. coli A and E. coli B. The genes will be inserted, right? And they will produce the chains of A and B separately. When it will be purified, when it will be purified, the chains A and B, they both will join. The chains, they both will join, and they will form an active insulin. Active insulin. So this is how the production of therapeutic drugs in context to insulin is occurring, right? Now, the second thing, Yeah, okay. Now I can see some of the response from students. Hi Jay Kumar. Hi Ayush Shukla. Hello Khan Shokat Ahmed. Hello Sam. Okay, good. Now the second application that we are going to see in today's class is formation of vaccine. This is the second application, vaccine formation. So this vaccine formation was done in context to hepatitis B vaccine. Hepatitis B vaccine, right? So this is the first synthetic rDNA technique, first synthetic hormone, first vaccine to be produced by rDNA technology. Always remember, because the first things are usually asked. So how the hepatitis B vaccine is given? There are three doses which are given consecutively in 6 months, right? So the production of hepatitis B vaccine was with the help of rDNA technology, hepatitis B vaccine. That was the first one. Now let us talk about the uh, third example of rDNA technology, that is DNA vaccine. Very important these days, and most of the vaccines, they are DNA based these days, and research usually is done for DNA vaccine. So what is a DNA vaccine? A plasmid with gene for a particular enzyme, for a particular antigen will be inserted into a cell. A plasmid with gene for particular antigen will be inserted into cell. Now, if directly DNA we are giving, it means it is capable of eliciting the immunoresponse, right? It will elicit the immunoresponse, but it won't elicit any kind of pathogenicity. So that now it is just eliciting the immune response, and it is non-pathogenic, because it is just a section of genetic material which is just forming the antigen. So it is very helpful, because there will be very less kind of uh, any harm because of this kind of vaccine, because it is just the use of the gene. So it will elicit humoral immunity, it will elicit the T cell immunity, and it will be highly helpful in cell mediated immunity, right? So three examples we have seen, that is production of insulin hormone, production of hepatitis B vaccine, DNA vaccine, and now erythropoietin production. Erythropoietin, erythropoietin production, and erythropoietin production, it will be directly helpful in what? Will be directly helpful in treatment of anemia, right? So production of erythropoietin can also be done. Then for treatment of neutropenia, what can be used? Filgrastim. It is used for treatment of neutropenia. Filgrastim. Then next, there can be formation of coagulation factors as well. Coagulation factors. Now, these coagulation factors, type 8 and type 9, they can be very well used for treatment of hemophilia. Treatment of hemophilia. Right? These are the applications we are reading for our rDNA technology. Now, next application is preparation of interferons. Preparation of interferons. Now, these interferons, they can be used in viral infection, and in cancer, and these interferons can also be formed with the help of rDNA technology. Then interleukin production. Inter Interleukin production. So this interleukin production can be very much helpful in renal cell carcinoma, right? Then production of growth factors. So epidermal growth factor can be highly beneficial in all kind of burns and skin associated diseases, right? Then there can be production of FSH, that is follicular stimulating hormone, and infertility can be treated. Infertility, infertility can be treated with FSH. Then LH, it can be used for ovulation and it can be produced in high amount with the help of our DNA technology. Then growth hormones in cases of growth failures can be produced. Human growth proteins can be produced. Then this gene therapy, which was possible only because of our DNA technology, this gene therapy, which was possible because of our DNA technology, was done for the first time for which disease? It was used for treatment of which disease? It was used for treatment of SCID, right? Then another application is DNA fingerprinting. DNA fingerprinting. So all the kind of medical legal cases can be solved with the help of DNA fingerprinting. Then from the starting, I'm telling you that the cDNA libraries or expression libraries can be made with the help of rDNA technology, right? And then there can be formation of transgenic animals. Transgenic animals. So which was the first transgenic animal to be formed? That was our Dolly sheep. Dolly sheep. So what all we have seen in application of genetic engineering, just do a quick revision. That is for the production of insulin hormone, then hepatitis B vaccine production, then DNA vaccine production, then erythropoietin for treatment of anemia can be made with rDNA technology. Filgrastim can be made with rDNA technology to cover up neutropenia. Then coagulation factors 8 and 9 can be made with the help of rDNA technology, which are very much helpful in treatment of hemophilia. Then interferons for viral infection and cancers, interleukins for renal cell carcinoma, for growth factors, FSH for infertility, LH for inducing ovulation, then growth hormones, human growth proteins. Then in gene therapy, rDNA technology was highly used, and the first disease which was treated was SCID. Then DNA fingerprinting is also association of rDNA technology and it is used for setting various medical legal cases. Then cDNA libraries to see which all the particular sequences in the genetic material are active in an organism, that is cDNA library production. Then transgenic animal production is also done with the help of rDNA technology. What are these transgenic animals actually? The transgenic animals are those animals, these transgenic animals are those animals which have got particular type of genes, and these genes are new to the transgenic animals. They are not actually the part of the transgenic animal genome system. So these genes. And for seeing various kind of mutations, there can be formation of knockout mice and knockout animals, and there can be formation of transgenic mouses, transgenic animals, gene knockout, gene knock-in. All these kind of things can be possible with the help of our DNA technology. So like this, we have covered almost every part of our DNA technology in detail. Maybe in some other class we can do the objectives and PYQs related to the topic. And I hope everyone who are watching presently live, they have liked the content and they have understood the concepts well. And they can do one thing, they can just write the topic which they want to study in biochemistry in the chat box, and we can just do some of the videos according to your demand. So you can just write the thing. Okay, thank you very much, Sam. And you can also write the topic which you want to study. Okay, so we'll be covering those topics. And in the chat box also you can write your query questions, so whenever I'll be going through my older videos, I'll be checking out. I've made lots of videos on molecular biology and enzymology and some of the techniques of biochemistry. You can just go and check out the things. Please start question discussion, ma'am. Yeah, in some other class we can start the question discussion. Here I haven't included, but everywhere in the theory I have given the idea that what kind of questions will be asked. So maybe in future we can do a video on the PYQs of RDNA technology. So just subscribe the channel and you will be getting the update when I'm doing the PYQ of RDNA technology. Okay, ma'am, complete genetic engineering. Sam is saying, ma'am, complete genetic engineering. Okay, we can discuss some other details also of genetic engineering, like the libraries, cDNA libraries, and all of the gene transfer technique in detail in the further classes. I'll be definitely keeping your suggestion in mind that you want genetic engineering in further classes. So subscribe the channel and you will be getting all the updates regarding the videos which we are making. And please don't forget to hit the bell icon for all the notifications, subscribe our channel, and please like my video and positively attend the class tomorrow at 10 a.m. and at 5 p.m. I'll be doing a class on heme metabolism tomorrow at 10 p.m. and a class at 5 p.m. So just keep tuned and we'll be meeting again in the next class. And if you are going for a plus subscription, use my referral code FB01 and you will be getting an additional 10% discount by using my referral code FB01. So please don't forget my referral code, that is FB01. It was very nice teaching you all. Thank you very much. Have a very good day and come back for the next lectures. Okay, bye.